And it's my pleasure to introduce Madeleine Ostwald from the University of California, Santa Barbara, who's going to give us an announcement about big bees. Madeleine, over to you.
Okay, thank you. Okay. So thanks for having me here this morning. As you just heard, my name is Maddie Ostwald, and I'm a postdoc at UC Santa Barbara, working with Katja Seltman on the Big Bee project. And so today I'm going to be talking more about this project and how I think Big Bee can be relevant to all of us as bee researchers. So the broadest goal of the Big Bee project is to enhance bee research opportunities through digitization and other efforts, particularly with the goal of understanding how bee traits drive responses to global change. So when we think about this huge question, how can we enhance global bee research? To me, there are several important themes. And one of the obvious answers is that we can always use more data. And that doesn't just mean launching new studies, but taking advantage of existing data deposited in natural history collections, digitizing it and making it publicly available. And we could do that by creating a centralized portal or database that hosts this data in an open access format that makes it easy for researchers to share and access bee data. And we can take advantage of all that data by developing new, exciting tools for quantifying different bee traits. And that in turn is going to help us generate even more data. So how are we doing this? So first, we're relying on the participation of 13 natural history collections across the US who are all digitizing their bee specimen data, as well as producing high quality images of their specimens, all of which are deposited in something called the Bee Library, which is a Symbiota portal. So there's a lot of things you can do on the Bee Library. You can search for species like here, Bombus vosnesenskyii. You can view all the occurrences of that species, and you can even generate an interactive map showing these occurrences geographically. So the Bee Library is live, it's currently usable, and it's growing every day. So as of the end of July, the library hosted over 2 million bee occurrence records, both from specimens and from iNaturalist, 87% of which are georeferenced, 39% of which contain images, which amounts to over 800,000 images, and half have been identified to species. So you can imagine that the research possibilities from this kind of data are endless. And as we're only in the second year of the project, the data here is still in its kind of rapid growth stage. And one of the really important features of the Bee Library is that we're populating it with a variety of images that can themselves serve as research subjects. So we're not only including standard label images, which can be used to quantify body size, but we're also including these high-res focus stack diagnostic images that will be used to visually describe focal features for species and genus identification. So if you've ever tried identifying a particular species from drawings of its characters and keys, coupled with like grainy photographs on Google Images, you can see how having these high quality exemplar images can make species identification a lot smoother and more standardized. And we're also including photos of male genitalia to assist in identification. And finally, we're generating these really cool 3D models of specimens, which will have a whole host of research applications, especially for morphological work. So a major aim of the Big Bee project is for all of this image and specimen data to be useful for this goal of quantifying variation in bee functional traits, and particularly in the context of global change. So when I say functional traits, I'm talking about traits like body size, sociality, phenology, nesting biology, pilosity or hairiness, and associations with other organisms like flowers or parasites. Specifically, we're hoping to achieve this in part by developing new tools for quantifying this trait variation. So I'll give a couple examples of what we're doing in this area. First, in one of the best studied bee functional traits, which is body size. So if you've ever measured bee body size, chances are you spent a lot of time either sort of wrangling a bee to sit on a microbalance or possibly spending many hours measuring intertegular span under the microscope or an image shay. So we've been piloting a new method for measuring intertegular span, which is a proxy for body size in bees, using this really cool community science platform called Notes from Nature, which is part of the Zooniverse. So Zooniverse has a really awesome dedicated user base of community scientists who help out on real research projects. And this has been especially powerful in the past couple of years with more research going remote. So if you've ever heard of Notes from Nature before, you've probably heard of it as a tool to crowdsource the transcription of museum records from images of labels, which is one thing that we're using it for. But they've also just launched a new really cool tool to take measurements from images. So we're using this to measure body size of our specimens. And it's really amazing because you can process a ton of samples in very little time. For example, in the first 2 weeks of launching our project, we had nearly 400 volunteers and 2,000 completed specimen subjects, with each of those specimens getting measured 5 different times by 5 different people to get a more accurate measurement. But because this is so new, it merits some thought about how accurate these measurements actually are. So we have a couple of undergrad researchers who are doing some analysis of how these measurements compare to measurements done by trained scientists measuring the exact same specimen under the microscope and in ImageJ. And then we're using that data to modify our instructions to Notes from Nature users and to learn how to clean the data output with the goal of fine-tuning this protocol. And this tool is already being used for research by one of the Big Bee postdocs, Colleen Smith, who's measuring body size across a group of common solitary bees and trying to understand if and how bee body size is changing over time and with different climatic variables. So that's one of the tools we're developing to help researchers study functional traits. A second one that I'll focus on today has to do with pilosity, which is bee hairiness. So we know that hairiness is a really salient functional trait when we're talking about pollination because it impacts the amount of pollen a bee can carry. And also when we're thinking about thermal tolerance, because hair has important insulative properties. However, pilosity tends to be really understudied, probably because it's so challenging to quantify. So traditionally, and still today, the best and really only way to know how hairy a bee is to count some subset of individual hairs on the body, or measure the length of hairs, or ideally do some combination of both. But this is, of course, incredibly labor intensive. And hairiness is also really complex. So within a single specimen, you can see huge variation in hair structure, hair length, and hair density across the body of a single bee. So a solution for this might be to look to some recent advances in computer vision and machine learning, which have developed really powerful pathways for identifying human hairs from images. So we're adapting some of this work to see if it will work for bees as well. So the pipeline is currently identifying bee hairs with an accuracy of about 75%, which is pretty good, but we're hoping to keep improving this number as we continue modifying the code and adding more and more ground truth images to the training data set. So ultimately, I'm hoping that this tool will help us to answer questions about the functional significance of pilosity by linking it to some thermal tolerance data that I've been collecting to see how hairiness impacts heat and cold tolerance across bee species, and whether this could even explain some broader biogeographic trends in the distribution of pilosity. So those are 2 really exciting tools that are currently in development, but we're interested in functional traits much more broadly than that. And ultimately, we're working on figuring out how to increase accessibility of trait data for all of these functional traits and many more. So for example, we have used specimen label data to deposit over 100,000 different bee interactions in GLOBI, the Global Biotic Interactions Database. We also already have a wealth of occurrence data available in the bee library that can be used to infer phenology. And then for me, at least one of the most exciting next steps is to see how we can better share behavioral data, especially related to sociality and nesting, which we're sourcing from the literature and from field observations. So with that, I'd like to just wrap up by encouraging you to think about the ways that this data might expand opportunities for your own research, whether by using occurrence data to inform species distribution models or models exploring changes in bee phenology over time, or perhaps tracking species introductions, or by using species interaction data to understand diet breadth in bees, for example. And finally, these images provide a wealth of opportunities to explore visual traits like body size. So for example, how does body size vary across landscape gradients like urbanization or elevation? Or potentially even looking at wing wear variation across seasons. And you can also look at interspecific variation in traits, for example, exploring the evolution of pollen collecting hairs in different taxa. So the possibilities are really endless, and I'm more than happy to chat with anyone who has more specific questions about whether there's data here that could help you answer your research questions. So thanks so much for listening, and thank you to all the many collaborators on this project. And this has, of course, just been a really fast overview, but there are a lot of other exciting things in development that I'd be happy to talk about with anyone who's interested. So please don't hesitate to contact me, and I'll take any questions now.
Great. Thank you for being so much on time. Jens Bokelar has a question. Is there a plan to extend this to the rest of the world and say to European collections?
That's a good question. I would have to sort of check with Katya about that. I think that would be like a really excellent next step and maybe beyond the scope of the current NSF grant. But so, you know, of course, there are some European specimens deposited in US collections, but nowhere near the diversity and the sort of depth of sampling we could get by collaborating with other collections. So I think that would be a really important next step.
From the chat, Benjamin Pinson has asked, what kind of supervised neural networks did you use to compare pilosity, and how many categories of sociality are you going to use?
Good questions. I would have to check about the specific kind of supervised neural network we used. I can tell you, at least now, that a lot of the base for this is a computer vision algorithm called Figaro, which was developed to identify human hairs. So that would be a good place to go to find out more about the specifics. It's a little bit outside of my domain, but I can also check and get back to you. And then how many categories of sociality I'm going to use is a really good question that I'm always going back and forth on. I think what I've kind of landed on now is that by including many different categories of sociality, for example, using Michener's categorizations, can then allow users to lump those however they decide is appropriate. So if you prefer to think of all, parasocial bees as social, and, just have, for example, just solitary social and eusocial, you can maybe filter the data that way, but giving people the option by including as much detail as possible in the database and allowing them to make their own inferences from that is kind of where I'm leaning now.
And thank you. And Meenakshi asks, do you have any inputs on how best to check for forager age, which probably relates to your wingwear comments?
Yeah, I think this really depends. This is really taxon specific, because in some bees, foraging effort is not always correlated with age. So it would depend on the species. But I think if you're making those inferences within the context of a single species behavioral repertoire, then I think there's a lot you could say about possible age of specimens from wingwear alone, if that's kind of what you're getting at.
We're almost out of time here, but Fabian has a question about, do you think you use your methods to take into account the morphological diversity of pili? Which ones can be innervated by receptor neurons? Oh my God, from a photograph.
Oh, that's a really good question. I hadn't thought about how we're going to distinguish the ones that could be innervated. But yeah, accounting for this diversity in hairs is really important. So what we're doing is trying to see how well these machine learning quantifications match up to human counts of hairs. And in doing those human counts, we're trying to account for diversity of hairs. So often there's, a layer of really dense short hair and then longer hairs coming out of that. Of course, some of those hairs are branched, some are unbranched. So we're trying to account for some of that complexity and see how much of that complexity is reflected in the overall machine learning output. So
thanks. It's time for us to go on to our next speaker. And